Hey everyone, with Trump 2.0 underway, along with the most orchestrated attack on science America has ever seen, you're probably going to be hearing a lot from me about the Orange Clown and his breathtaking science illiteracy. Today's installment is about some alleged blue-haired, ultra-woke transgender mice and the millions of dollars that Biden wasted on them. Let's get into it. So this all started when Trump addressed Congress on March 4th, vomiting up a long list of alleged extravagant expenses that the previous administration put upon the poor, unwitting taxpayers, gloriously uncovered by President Musk and his Dodgeball team. The whole speech is a circus sideshow from top to bottom, and we could dig into virtually every single one of his ludicrous claims. But let's focus on this one.
$8 million for making mice transgender. This is real.
No, it isn't real. If you made a list of not real things, this would be pretty high up on there. Transness is not something that applies to mice. Nobody is making trans mice. He's just saying trans because transgender issues is one of the topics in the pathetic culture war he's waging that riles up his idiot supporters. Naturally, thousands of intelligent people called out this utter bullshit, mocking him relentlessly for saying such an idiotic thing, and we will explain in more detail than you want to hear how idiotic it is in just a moment. But first, this is how the President of the United States decided to respond to thousands of scientists fact-checking his ridiculous claim. Wrong, you guys. Biden totally did it. Those fake news losers at CNN tried to fact-check me, but I was right, as usual, because I'm always right, because I'm such a smart special boy like my mommy always told me. You guys, this is not a tweet. This is not a blog post. This is from whitehouse.gov, the official website for the White House. This is what America has become. The man who holds the highest office in the country, and arguably the world, talks like a bratty teenager in official White House communications. We're halfway to idiocracy, and I'm not being even remotely hyperbolic.
The President of America! I got a 3-point plan to fix everything.
Department of Government Efficiency. DOGE.
Getting chills? Well, tough shit, because it's a fact, you guys. Biden spent $8 million to turn mice trans. You know, those dirty transgender experiments that are totally a real thing. Then he lists the grant amounts and titles of the studies, the end. Well, that's very convincing, Mr. President, but first of all, $8 million is a completely negligible fraction of the $6 trillion federal budget, especially compared with the tens of billions of dollars sent to Israel over the past year and a half for the sole purpose of murdering Palestinian children, a convenient omission from your little list. But beyond that, some people actually know what all of these words mean, even if your cronies don't. I mean, maybe the 19-year-old DOGE staffer who goes by the name Big Balls knows what some of these words mean. He seems like a real wunderkind, but I wouldn't bet any of his rich dad's popcorn money on it. Anyway, they were kind enough to attach a document linking to all these proposed studies, so let's go through every single one of these 10 scary grants with trans in them and explain what they're actually about. Spoiler alert, there isn't any waste, fraud, or abuse in any of them, so let's teach Trump a little science. First up, molecular mechanisms of hormone-mediated sex differences in wound healing. If someone took 5 seconds to read the first sentence of this grant, they would see that the first thing they address is that the cost Americans as a population spend on treating chronic wounds, or those that don't heal within 6 to 12 weeks, something which affects 1 in 50 people, is roughly $100 billion per year. Doing the math, $340 million over 50, $100 billion over all those people, that's 15 grand apiece. Assuming there is no healthcare mechanism to assist all these individuals, something which Donald Obamacare is the devil Trump truly wants to be a reality, that means 1 out of every 50 of us might have to spend almost $15,000 out of pocket. Sounds like this research could reduce the cost and improve the efficiency of treatments that millions of people will need for a collective tax investment of a measly $450,000. They also mention other afflictions that this research could have relevance towards. Weird, nothing about transgenders yet. Then they summarize what we know so far, that there seems to be a correlation between someone's sex at birth and their ability to heal wounds. Apparently, men, on average, are worse at healing wounds than women. Interesting. They go on to say that researchers have noticed testosterone and estradiol might specifically have some important role in these processes. But things get really confusing because it also looks like they might not be doing the same thing when examining men versus women. It would be better experimental control if we could study the effect of men taking testosterone or estradiol versus not taking them, and also the effect of women taking testosterone or estradiol versus not, so that we can figure out exactly what is going on here. They mention that examining transgender individuals would be a good idea because sometimes this is part of treatment for trans individuals. They are talking about controls. They want to utilize the fact that trans people exist in order to control for sex, to understand what a hormone does in an XX individual versus what it does in an XY individual. They don't even know if the effect of estradiol is different between sexes. They want to do that experiment, and of course we always test such things on mice first. They talk about testosterone affecting the immune system and how they want to learn more about that interaction in XX versus XY animals. Nothing so far about asking the mice their preferred pronouns. They are just looking at how exogenous, meaning externally applied, hormones might improve or hinder wound healing that they think is acting through the immune system, and how it works depending on whether you have XX or XY sex chromosomes. That's it. They used the word transgender once in a sentence just to say, hey, it might be a good idea to take a closer look at trans people getting healthcare because it might give us useful data to help understand wound healing and save people money. But that's too triggering to Sir Groper Cleveland, so we won't get that benefit. By the way, whichever brain-dead intern threw together this cute little document for Trump decided to highlight the most triggering quotes in the grants. Apparently, they were the most upset about, we developed new models and showed that testosterone limits wound healing in large animals with different effects in XX versus XY animals. Controls are super woke and lame, guys. Chronic wounds are much better. Kill the study. Also, we should remember that grants in science are very competitive, with an application success rate of roughly 20 to 25%, and reviewed by a panel of competing scientists within the relevant field who would absolutely love to take a giant shit on your research proposal if they see a discrepancy in your logic. If these grants sounded like an absolute waste of money, they would get trashed. Moving on. A mouse model to test the effects of gender-affirming hormone therapy on HIV vaccine-induced immune responses. Trigger warning, this grant contains scary words acknowledging that trans people exist. If you have the testicular or ovarian fortitude to get past the scary bits about trans people receiving healthcare, since they are in fact people, then we can move on to the science. Like the previous grant, they mention how sex hormones such as testosterone appear to influence the immune system, but those pesky sex chromosomes keep adding noise to the data. They say they could sort out the noise from the data if we could just control for sex differences. They specifically are interested in how vaccine treatments induce differential immune responses between the two sexes, and looking at gene expression differences within a sex could be an interesting way to generate hypotheses about what might be going on. Wait a minute, weren't there people in real life complaining about vaccine efficacy needing more research? I seem to recall a bunch of people whining about a recent vaccine being experimental and how we were all guinea pigs and they killed millions of people. For those people to be consistent with their wrongness, you'd think they'd be interested in experiments that could inform us about how we can prevent negative responses to vaccines, yeah? Especially if it looks like there might be sex-specific differences. Oh, but we would have to be able to control for sex. So their experimental model is essentially apply some amount of hormone to mice of a specific sex, administer the vaccine, and look at the genes that respond. It sounds like if there are treatments outside of gender-affirming care that involve hormonal treatment, and there are, it could inform us about vaccine safety that affects those completely non-trans people as well. Crazy, huh? The most triggering parts of the grant, according to, let's call the intern Rory, were apparently, we propose to develop an animal model of feminizing hormone therapy to study the effects of estrogen anti-testosterone therapy on HIV vaccine-induced immune responses. And we will develop a mouse model of XHT, cross-sex hormone therapy, that recapitulates clinical hormone therapy for male-to-female transition in humans. See this word model? In science, developing a model means to create a representation of a system for the purpose of making predictions. We don't make weather models by creating literal isolated weather systems. We don't actually recreate the early universe at the Large Hadron Collider. Likewise, in biology, we don't literally create transgender mice. They are models used to mimic real-life phenomena and can be used to ask a variety of questions depending on the limits of the model. Again, there are multiple good scientific reasons for studying hormonal effects within a sex, and transgender healthcare is just one extension of direct application from this model. Apparently, it's just too much to handle that trans people are one of the immediate beneficiaries of this research, so let's just ignore research that could help anyone who receives any vaccine. Very on theme for His Majesty. Number 3. Reproductive consequences of steroid hormone administration. They start off talking about how there are few studies on the effects of cross-sex hormones on fertility. This might be a good point to remind everyone that there are numerous health issues that stem from an imbalance of endogenous hormones. So, a study like this could easily pertain to what we know about a multitude of hormonal disorders, including polycystic ovary disorder, which is thought to be due to an increase in male hormone production in women, affecting around 10% of the female population. They comment on the lack of data on transgender individuals. Again, let's remind ourselves that there is a scientific reason to want to control for sex when studying hormones due to innate differences on average, and trans individuals just so happen to fit the bill as perfect patients for observing these effects in humans. By extension, controlling for sex in mice provides a powerful experimental model to test hormonal treatments, with the authors stating, "This model provides a powerful tool to clarify the effects of T therapy on reproductive phenotype and function in a manner not possible in humans." Despite all the things we could learn about fertility in men and women, the fact that the repercussions could include providing healthcare to trans people is much too scary. So we are now banning controlling for sex. Sorry to the 1 in 10 men and women who struggle with infertility. Donald says go fuck yourself. According to Rory, the key triggering statements in this grant were, the impact of long-term cross-sex hormone therapy on reproductive health is largely unknown, particularly in transgender men, female to male or FTM. And none of the existing animal models that address the effect of androgens on reproductive function in females are directly applicable to the clinical paradigm of cross-sex T therapy in transgender men, or GnRHA-T therapy in transgender adolescents. To address this knowledge gap, we have developed a mouse model to mimic T treatment for FTM gender transition. So to summarize, there is a knowledge gap, and current models cover X relevant health issue, and they designed one that covers X+1 relevant health issues. Apparently, increased efficiency gives Donnie D. Cups the runs, so let's cut him some slack. Moving on. Cross-sex steroid therapy and cardiovascular risk in the transgender female. Again, we need to get past the scary words talking about trans individuals and get into the actual science. They mention that their observations that individuals that were born male and receive hormones for gender-affirming treatment have an increased risk for cardiovascular disease. So presumably, there is a molecular mechanism underlying this phenomenon. The words talk about the benefits to trans people, but by understanding the science associated with it, this research can easily apply to cisgendered people. If we understand the effects of hormone X in males versus females on cardiovascular health, we can come up with better treatments for anyone with cardiovascular issues. They end with how this grant will improve their skills, which is a sign that it was written by a student. So they defunded a graduate student and their science that would contribute to knowledge about cardiovascular disease because it contains words about transgender people. But who cares about people with cardiovascular problems? If you eat nothing but McDonald's every day, you deserve to die, right? Rory found this grant particularly triggering, as him highlighted several sections of it. The Alexander Laboratory has developed a novel model of feminizing hormone therapy in the male rat that involves administration of E2 to mimic physiological levels observed in age-matched female rats in conjunction with androgen suppression. In other words, they control for sex when applying hormones. A critical need involves the use of innovative animal models to provide reliable risk assessment and in-depth investigation into mechanisms that contribute to increased CV risk in adult transgender females, individuals that undergo GAHT. Yes, this would benefit trans women directly because that is the most direct application from the model. Oh no, then they won't die as much. It will also add to our knowledge on mechanisms underlying cardiovascular diseases in general, and in a way that can only be done by controlling for sex before applying hormones. Control's bad. Trump smash. AIM-1 will test the hypothesis that the shift in the hormonal milieu in gender-affirming hormone therapy in a rodent model of the trans female rat is associated with increased end organ damage and cardiovascular risk that is further exacerbated with pre-existing chronic disease. AIM-2 will test the hypothesis that increased end organ damage and cardiovascular risk in transgender females that undergo gender-affirming hormone therapy involves an estradiol-induced male sex-specific effect on the renin-angiotensin system. Translation: AIM-1 tests whether a hormone treatment cocktail used in a real-life treatment will influence cardiovascular disease. They simply use an intervention that contains relevance in real life. Scientists try to squeeze as much out of their data as possible. Why wouldn't they use a similar hormone treatment that could be applicable to humans? That's not fraud, that's called efficiency. It's starting to seem like they just did a search for the word transgender, read literally nothing else, and pulled the money just to add an item to Trumpelstiltskin's dumb speech, huh? Next up, gender-affirming testosterone therapy on breast cancer risk and treatment outcomes. This one starts with the obvious direct benefit to trans individuals. Their experimental design of controlling for sex is also ideal for looking at effects of miRNA and testosterone on mammary gland development, carcinogenesis, and breast cancer treatments, as they point out in the very next sentence. This sentence could confuse a lot of people, so imagine how the Trump team took it. MicroRNAs are small, non-coding RNA molecules that regulate gene expression. Carcinogenesis is when normal cells become cancer cells. The rest you probably know. Anyway, by examining the effects of testosterone levels in females, we can learn about how breast cancer is affected by differences in testosterone levels, which could be beneficial to the 1 in 8 women who are at risk of developing breast cancer. You'd think Trump would give tits a pass, but not this time. They make a note that the mouse studies here are particularly useful compared with human studies because the rate of aging in mice is much faster, while breast cancer development appears to be very similar in mice, meaning they can learn about breast cancer at a much faster rate by using mice studies. And if testosterone has an interaction with the development of breast cancer, wouldn't it make sense to learn about this interaction so we can potentially make new drugs and learn about negative interactions with existing treatments and the various types of breast cancer? Why would we not do this? Oh, because it could also help trans people? Maybe it makes sense if you ingest bleach, eh, Donnie? If they had been able to get through one paragraph, they would have seen these knowledge will have direct implications for understanding BC risk and open up new avenues of treatment for cisgender men and women as well. Oh well, it's funny how Diaper Dawn talks a big game when it comes to helping kids with cancer and is happy to use kids with cancer in a political stunt, but then actively defunds research that can help cancer patients, huh? What's next? Microbiome-mediated effects of gender-affirming hormone therapy in mice. They specify that we don't know how certain hormone treatments that are provided before puberty affect skeletal development. This includes those used for non-gender affirming care, prostate cancer, endometriosis, and central precocious puberty. They want to know how blocking or inducing certain hormones can affect skeletal development. The microbiome has also been shown to be a factor in skeletal development, so they design experiments that include fecal transplants to disentangle effects from either mechanism. Would you want to know about side effects of a treatment on your child if they needed hormone therapy before puberty? Would you like to help ensure the health of children with cancer or reduce pain in young women with endometriosis? Mango Mussolini says no, let's not, since it will also benefit trans people. What's next? Androgen effects on the reproductive neuroendocrine axis. This one should be clear as fucking day, but let's break it down anyway. Androgens are hormones typically associated with the development of male characteristics in humans, including testosterone. Although present in much lower quantities, females still produce testosterone and other androgens endogenously. Some females produce more or less on average. Levels fluctuate throughout the menstrual cycle and typically decrease with age. First word down, let's continue. Androgen effects on the reproductive neuroendocrine axis. The neuroendocrine axis refers to the hypothalamus, anterior pituitary, and an end organ system through which hormones are released based on a multitude of inputs received from throughout the body, creating hormonal feedback loops that can regulate physiological functions, including body weight, thermoregulation, hunger, fatigue, and reproduction, to name a few examples. The grant title refers to the reproductive aspect of the endocrine system. Putting it together, the title of the paper suggests they want to look at the effect of male-associated sex hormones on the reproductive aspects of the endocrine system. Once again, females also produce androgens, which act through the endocrine system and ensure normal reproductive function. They propose looking at the effect of androgens on a specific type of cell called hypothalamic kisspeptin neurons, which produce surges of luteinizing hormone involved in a complex cascade of events that ultimately affects reproductive capabilities in men and women. The experiments they propose are to monitor reproductive aspects of trans men receiving healthcare, because they often are treated with exogenous androgens as part of their healthcare, which we can then compare to women who are not treated with androgens. The other experiment involves using a transgenic mouse model, presumably by genetic ablation of kisspeptin neurons via KISS1 knockout, though it's not specific in the abstract, to determine to what extent the effects of androgens act through the kisspeptin neurons. The results from the experiments laid out in the grant could be informative to menstrual health, reproductive health, and cancers acting through this system, in addition to trans health. It's important to note that just because a grant is funded through an initiative that aims to address knowledge gaps in the literature, this doesn't mean that its benefits are entirely limited to the scope of the initiative. As we went over in the previous grants, yes, of course they benefit trans people, but they also benefit cis people because the models they've described thus far provide a framework to make predictions in multiple forefronts of science. That means the experiments were well designed. Read for yourself. Indeed, high levels of exogenous androgens fundamentally contribute to reproductive disruption seen in otherwise healthy women and transgender men, female sex individuals taking high levels of androgens. Oh look, that's pretty neat. They say right in the grant that it's applicable to both cis and trans individuals. I guess the E for efficiency in DOGE really just means Control F search trans and delete without knowing what the fuck you are actually deleting. Great work, Elon. Let's take a look at what triggered Rory on this one. This proposal includes clinical studies of transgender individuals and the effects of androgen treatment on their reproductive health. Yep, that's the fucking experimental design, genius. It's like getting triggered by someone explaining the ramps and pulleys in high school physics class that will test Newton's laws. We test this hypothesis in two complementary aims that study the role of high exogenous androgens in both a clinical setting in transgender male-female sex, human subjects, and corresponding transgenic female mouse models. Using complementary experiments to corroborate results just makes it good science. It's so painfully obvious that they just grab the statement they think makes the tangerine dream appear vindicated, when in fact they just look like complete morons that don't understand what's being described in the slightest. But wait, there's more. GHB toxicokinetics, role of sex hormone-dependent monocarboxylate transporter regulation, and potential for altered overdose risk in transgender men and women. The researchers state that preliminary data show that toxicokinetics regarding GHB, gamma-hydroxybutyrate, commonly referred to as the date rape drug, are altered in the presence of female sex hormones. This is supported by previous research showing that hormonal changes associated with the estrus cycle, or mammalian fertility cycles generally, may act through monocarboxylate transporters in the kidney, which are the same transporters important for GHB clearance. The research questions they want to test are: 1. Do individual sex hormones affect GHB clearance? 2. Does testosterone make GHB overdose more likely? The only way to address these questions properly is to control for sex before applying treatment. Not controlling for sex will decrease efficiency of the science by adding unnecessary noise to the data, making analysis more complicated and wasting money, assuming this research is possible at all without these controls. If you can't understand that and are still fuming about trans mice, you're a dumb bigot, probably of an orange tint. The quotes that Rory pulled from this one really show how obsessed these idiots are with their caricature of transgender culture. Gamma-hydroxybutyrate, GHB, is a popular drug of abuse utilized at raves. Yes, people abuse drugs at raves, and also in offices, homes, streets, many places. Shall we study the drugs then? No? Use of GHB has been increasing in the LGBTQ community due to the prevalence of a phenomenon referred to as chemsex. Wow, some people do drugs, some people have sex, and some people even do both at the same time. Let's all clutch our pearls about the horrors of the real world. If Prezi Pumpkin could stop fixating on chemsex, he might understand that this could also inform us on how to treat date rape victims generally. If we know that certain individuals can have worse reactions to the drug, and we know why, we can design treatments to combat the overdose. Still more, you guys. Gonadal hormones as mediators of sex and gender influence in asthma. This is probably the dumbest inclusion on this list. It is literally just proposing that we look at sex differences on asthma phenotypes. They have data showing that the inflammatory response in asthma might be triggered by estrogen. Women who go through fertility treatments often take estrogen. This is not a crazy, wacky idea. They want to do experiments to look at the effects of estrogen on inflammatory mechanisms in mice with XX and XY chromosomes. They're not making transgender mice. They are looking at the inflammatory response in male mice and in female mice with or without exogenous estrogen. It's mind-boggling how simply having the word gonad in the grant frightens these toolboxes to the point that it depletes all rational thinking. They don't have the brain power to get past the scary words. Rory even specifically pulled out the statement, "We expect that our studies would serve to develop potential sex and gender-specific treatments and recommendations for dosage of therapeutic agents to treat and prevent asthma in cis and transgender women." It literally says cis and trans women. So I guess they just don't give a shit about women in general. Who cares about their asthma, right? We need the money to pay for Elon's super cool exploding rockets that load the atmosphere up with metal oxides and nitrogen oxides, plus all the child support. Finally, we get to the last one. understanding how chromosomal makeup and cross-sex hormone administration affect wound healing in mice. Reports had been floating around that transgender patients had worse healing outcomes than other populations. Additionally, this pattern doesn't seem to be equal between sexes. From this observation, they proposed that testosterone might inhibit mechanisms of wound healing. This grant wants to observe trans patients undergoing hormone therapy and complement it with a mouse model. Basically, the same fucking thing we've been saying this whole time. The researchers are just doing the best experiment to figure out what hormones do to the body without giving a shit whether it involves trans people or not. It's just an element of experimental control. But the best part is the last sentence in the grant, which they conveniently did not acknowledge and probably didn't even read. By filling this knowledge gap, this research may ultimately offer improved wound healing and personalized medicine therapies to not only transgender patients, but all patients. But who cares about all patients, right? That doesn't sound like an important demographic to the Donald or any of the bloodsuckers in his inner circle. So that's it for the 10 woke trans-loving grants that Trump canceled to save a few bucks. There are a few obvious takeaways here. First, none of them wanted to make trans mice. Second, all of them referred to trans individuals as a way of controlling aspects of hormone administration within a specific biological sex in order to see their effects on a particular aspect of animal physiology. And third, all of them outlined studies whose results would benefit all humans, not just trans people. Of course, we should also note that no matter how angry it makes conservatives, trans people do exist, making up around 1% of the population or more, and they matter just as much as cis people. So even if the studies were actually only applicable to trans people, which they aren't, it still would merit spending a few $1,000,000, which again is completely negligible in the grand scheme of government spending. Trans people deserve to benefit from research that they pay into through taxes, just like cis people. Given the total NIH budget of $48 billion, I think we can allow this 0.0002% of NIH grant funds to benefit 1.6% of the population. On a scientific level, the arguments used to deny funding for these grants that have already been approved by scientists as good use of tax dollars are tired and pathetic. We've heard these types of caricatures before. Sarah Palin, vice presidential nominee for John McCain in 2008, made comments about fruit fly research being a waste and taking money away from disability research. This idiot doesn't know that there is a Drosophila-based Center for Research into Autism at the University of North Carolina, which has provided decades worth of high-quality and groundbreaking research, including the discovery of genes being transmitted via chromosomes, the role of Hox genes in embryonic development, and molecular mechanisms of circadian rhythms. Many scientists have written extensively on why this type of ignorance is dangerous. Other examples of silly-sounding science weaponized by clueless politicians include the shrimp on a treadmill fiasco, which was used as justification to decrease funding to science, even though it had important implications on the effects of water quality on wildlife survivability. Researcher Lawrence Tabak was under fire from critics claiming that tax money was wasted sending toenail clippings through the mail. His response encapsulated the ignorance of such critiques in a quote, stating, "What's scientifically sound and indeed cost-effective to collect biospecimens for cancer research was twisted in what was intended to ridicule an important life-saving research effort," which is basically the same playbook being used right now to defund science by claiming that scientists are making transgender mice. The lesson that I desperately wish Trump and his peanut gallery would learn is that you can't make knee-jerk judgments about scientific research without understanding the science. Some of the most groundbreaking science sounds silly when reduced to one-liners. Studying worm vulvae led to understanding retinoblastoma. Looking at how green algae phototax in response to light led to the discovery of a suite of genes that are involved in the rare developmental disorder Bardet beetle syndrome. Looking at how algae swim led to insights into lung diseases, including primary ciliary dyskinesia. In science, we call these phenologues, or phenotypes that may have no relevance between organisms, but derive from evolutionarily conserved molecular mechanisms. These can lead to funny-sounding studies, but it is a valid and successful approach to studying disease mechanisms. Weaponizing provocative one-liners like transgender mice to stir up drama doesn't just affect science related to transgender individuals. It affects most of the research that we all want and directly benefit from. Oh, and it makes you sound like a fucking idiot, Donald. Just a tip. Thus ends the story of Trump and the woke trans mice, a cautionary tale that in a normal timeline would be getting Trump laughed out of office. But instead, we have to endure this sheer insanity on a near daily basis for an unspecified duration of time. What will the next debacle be? We'll just have to wait and see. Until next time.